You want to hit record for me again? All right, you got it, girl. All right. So membrane transport. Some of this you got a preview of if you've already had lab this week. If you have not, then you'll get a re-summary of this tomorrow. So this will be really, really reinforced for you this week, which is really nice. So membrane transport. When we're talking about membrane transport, we're really talking about getting things across the plasma membrane. Okay, so remember, when we were talking about the plasma membrane, up to this point, we really talked about it as a protective barrier. Okay, its main goal has been to keep things out. Okay, all that nasty bacteria, all those nasty viruses, any kind of danger. Its goal was to stop it. Okay, but remember, we also said that our cell is a living thing. Okay, so it needs to eat, it needs to breathe. Okay, it also is going to make cell poop, so it needs to get rid of that. Okay, and it's also going to need to breathe out. So we need some way for those things to be able to cross that membrane and do so safely. Okay, so this is really what membrane transport is all about. Getting things across our plasma membrane safely when we need to, but still being able to keep all the garbage out. Okay, so the first thing we're going to do is sort of review what our plasma membrane looks like, and this is going to be, remember, we always start on some solid footing where our platforms. So this is going to serve as that because we've really touched on our plasma membrane in almost every unit, right? We built it in our biomolecules. We revisited it in our tour of the cell. So we're going to revisit it here because it's going to play the biggest role in what decides what gets across and what doesn't. And then we're going to look at how things are getting across the membrane. And there's really two major groups for this. Passive transport, okay, and so when I say no charge, talking about fees, right, charging a fee. And remember, the only currency we really have in the cell is ATP. So passive transport is not charging a fee. It's not charging, gracious, charging ATP. And then if we get to it, we're going to talk about active transport, which is going to start to charge or cost us ATP, right, which we remember we made in our last unit using the mitochondria and the chloroplasts. Okay, but let's start by looking at our membrane structure. All right, so a lot of this is familiar territory. So remember, we said our goal here was to allow some things across, the things we want, food, oxygen, okay, to allow things we want to get rid of out, okay, excretion, aka cell poop, okay, we want to be able to exhale, but we still want to keep out all those danger materials, bacteria, viruses, invaders, abrasion. Okay, so the cell plasma membrane is the one making these quote-unquote decisions. Okay, it's still acting as a protector. Okay, so this is what we mean by selective permeability. Okay, it's choosy. Okay, it's being choosy about the things that it lets in. And of course, permeability being lets across. Okay, as we'll find, not only is it going to let some things like food in and danger not, but it's even easier for some things to get across than others. Okay, so we have a choosy membrane, or picky. Okay, so selective permeability. It's literally going to select which items can go across. Okay, by the nature of its own design, of course. Right, there's no gatekeeper sitting there pointing and allowing. little membrane gnomes. Okay, this feel okay? Here's some solid footing, right? Most of this isn't new. And most of this, even now, is going to be some new terms. But everything that we're talking about is stuff we already know. Okay, so let's look at our membrane. So remember that all of this is made out of phospholipids. So if I look at my image on the right, I remember, oops, definitely drawing. I have phosphate heads on the outside of my membrane. I just really am bad at ease, aren't I? Okay, so these are shown in gray in my image. And so remember, my phosphate heads are a phosphate surrounded by 4 oxygen. So remember, when I have oxygens, that means I'm polar. An oxygen starts with an O, both, right? So that means I'm polar. That's the way I remember that. See that question come up a lot in your exit tickets. Okay, so oxygen is what makes something polar. Okay, so remember, if you see a lot of any structure that has oxygen, phosphate heads, hydroxyls, carboxyls, all of these compounds have a lot of oxygen. So those groups are polar. Okay, remember that polar things play well with others. So they're hydrophilic. So some of the things they play well with is water. So this would be outside the cell and inside the cell. Conversely, we have our lipid tail, right? That's this business here. These are made of hydrocarbon chains. Okay. There's no oxygen on these guys, right? So these, shown in yellow in this image, are very nonpolar. Okay. Remember, these don't play well with others. Our little tails tuck themselves together. Tails tuck. Okay, they're scaredy cats. I'm afraid. Okay, remember, that's what's made this little closet here, which is what creates the protective barrier. Okay, we knew this already, though. This is what we built in our biomolecules lecture. So right now, we're really just dusting off some old knowledge. It just feels like it's been forever since we talked about it. So really, the key feature here that's brand new is our term. Amphipathic. Okay. Amphipathic just means we're going to put a definition to what we already knew. We have a hydrophilic section and a hydrophobic section. Amphipathic means you have both. Just like ambidextrous means you're both right and left-handed. Amphipathic means you're both hydrophilic and hydrophobic. Okay, so this amphi prefix means having of two, or having of both. In this case, our both is hydrophobic and hydrophilic, which is kind of weird. Okay, this platform feel good? Well, that didn't come out as English. This platform feel good? Okay, nice familiar plasma membrane territory. Okay, good chance to review our terms: polar, hydrophilic, hydrophobic, hydrocarbons, and all of that business. All right. So that leads us to start our conversation about how do we actually then take this information and use it to move things across a membrane. So let's start by looking at passive transport. Okay, so let's start by just looking at passive transport in general, diffusion of which is one of the types. No, I'm drawing. Okay, if I'm going to do any kind of passive transport, okay, the key feature here is these first two bits. Okay, moving down gradient. Okay, so what does moving down gradient mean? So if I look at my image, okay, the idea here is that really anything doesn't like to be crowded. Am I right? Okay, so this is more so now, okay, but was really always the case. When you walked into the room, okay, you probably never sat in the seat that was around a whole bunch of smooshed together people, right? You tended to opt for the seat that gave you a little bit of elbow room. Okay, and that's the case with molecules as well. So if I am looking at the left side image, this is called high concentration, right? Because there's a lot of individuals in a tiny space. They're all smooshed together. They don't have any elbow room. Okay? The right side of my image is low concentration. They're nice and spread out. They all have a lot of space. If I were to walk into the DMV, I'd want to sit on the right-hand side, right? Okay, I don't want the weird guy breathing on me. Okay, so the goal is if I was stuck here and I had the option to move, okay, I would want to move from the smooshed space to the emptier space, right? Okay, we all want a little more space. Okay. So that's what we mean when we say we're moving down gradient. Okay. We're moving from an area that is high or heavily smooshed to a space that's low or more open. Okay, until we spread more evenly. Okay. The goal, of course, ignore that one. Oops, still childlike writing. Is to reach equilibrium. As the name implies, everything being equal, right? So everyone has the equal amount of space. Okay, now that doesn't mean nobody's moving around. Okay, we've all been in the DMV or in a kindergarten classroom. People still get up and move, right? I want to go sit by Hank. I want to go sit by Jeremiah. Okay, people still get up and move. Okay, but there's no directed movement. Okay, there's no benefit to necessarily sitting on one side or the other. People are just sort of randomly exchanging places because they'd rather sit in one space or another. Okay, so that's what equilibrium is. And the key is things are still moving around. A stop cell is a dead cell. Okay, just like anything that doesn't move. If you and I stop moving at all, we don't move, we don't breathe, we dead. Okay? Same thing for our cells. So the molecules are still going to be moving, they're just randomly changing seats like kindergartners. Okay. Everything I've described up to this point is just general passive transport. Okay, so on Friday, what we're going to do is talk about what exactly makes diffusion different, as well as all of the other types of passive transport. Okay, so what sets these different types of passive transport apart from each other, and why do we have more than one kind? Okay. So for now, we'll just make diffusion the teacher's question. Okay, so for Friday, we'll work on this a little more. You'll also have a chance to ask questions about the exam.